Alright guys, so here we are. I took a little hiatus, but we are so back. And talking about a really interesting topic today. I think it's really worthwhile going over since the head of HHS has no respect for the scientific method and vaccines might not be around because of him. So I think it's important that we just appreciate what vaccines really do for us. In this case, I'm going to be talking about a seemingly simple question, which is going to take roughly 40 minutes to get to the answer. So if you don't want to watch a lengthy video about how vaccines work, maybe don't watch this one. But yeah, today's question of the day is, why is aluminum in vaccines? And so just before we really get into it, just a quick review of some vaccines we've had in the past, or different types of vaccines. We initially started out hundreds of years ago using attenuated vaccines. And so what these are, basically, in this video, I'm just going to refer to, there's many different ways you can get sick. There's bacteria, viruses, parasites, fungi, a lot of different ways you can get sick, but for a case of this video, I'm going to lump all of those under the name pathogen. So basically, you take a pathogen, you weaken it or kill it, and you re-inject it back into your body, and you create antibodies towards it. That's cool, but one drawback is if you're immunocompromised, there's still a chance that you can get sick from the vaccine. There's actually cases when we had polio epidemic or outbreak where this attenuated polio vaccine wasn't fully attenuated and some people just straight up got polio. So from there we were like, hey, maybe we don't even need the full thing. Let's just take a toxoid, or just like a piece of the actual pathogen from tetanus, for example, or purify a specific protein that's unique to that pathogen and put in a vaccine. Since then, we've also created vaccines from virus-like particles or attaching specific protein polysaccharide conjugates, and most recently we have the mRNA vaccine, which I'm sure we're all aware of. And I'm not really going to go into how this mRNA vaccine works, but just a general background of vaccines have been around for quite a while, although we still don't really know fully what's the best way for them to work. There's new methods being tested, and that's another thing I'm going to mention in this video. Is this is by no means a comprehensive view as to how vaccines work. This is merely going to be me trying to give lay people somewhat of an understanding as to why aluminum is in vaccines, since it doesn't seem very, I guess, common sense just looking at the statement aluminum is put in vaccines. But aluminum is in a vaccine because it causes an increased vaccine response. In this case, the aluminum is an adjuvant. So there's many different types of adjuvants, but aluminum is one of them, and the title, why is aluminum in vaccines, sounds the most clickbait, so that's what I'm going with. But there's many different types of adjuvants. Aluminum can be one of them. And what you see is that if you don't have adjuvants in vaccines, you have a lesser immune response than if you have a vaccine with an adjuvant. And it really isn't clear from this figure why you need aluminum in a vaccine. And so that's what I'm going to really go through today. And what I'm going to take you through really is what happens generally when you get sick, and how is that process hijacked when you're making a vaccine. So in order to talk about the immune system, let's just quickly talk about where it comes from, I guess. And all of your immune cells are coming from blood stem cells. And this stem cell will separate into two lineages, the myeloid lineage in the lymphoid lineage. This myeloid lineage will create cells like red blood cells and platelets, which is what you generally think of when you think of blood. But then there's also myeloblasts, which these create granulocytes, which generally, apart from neutrophils, are only really targeted towards parasitic infections, so I won't really mention them today. And then this lymphoid lineage is what is found in the adaptive immune response. So when you think of antibodies, you need a lymphoid cell to potentially create B cell, then T cell, NK cell. There's really a lot of complexity to this, and showing here a more complex, I guess, flow chart of this stem cell lineage. So from this blood stem cell, this pluripotent hematopoietic stem cell, depending on what molecules it's around, dictates what type of cell it'll become. So in this case, looking at this image here, if you're around IL-3 and GM-CSF, you're gonna go from a hematopoietic stem cell to a myeloid stem cell, and you can't go back. Once you differentiate down, there's no going back up. And basically what I'm trying to show here is there's a lot of different cytokines. These cytokines are signaling molecules which can influence what type of cell it is differentiated into, and is also used for signaling, which I'll mention later on. But just a main point of this diagram is further to note the immune system is very complex, and in no way am I saying this is a very full view of the immune system. But let's look at what first happens. Like, you get a pathogen, what happens? Are you creating antibodies right away? No. And the reason for that is, I'll mention this later, but it takes a lot of energy to make antibodies. So your body has a pretty foolproof system of getting rid of pathogens without ever needing to make antibodies, and that's by using complement activation. So what happens is you have a bunch of these complement proteins in your blood. So one of them is called C3. And when C3 is just floating around your body, it can get cleaved into C3b and C3a, which I'll talk about in a sec. But C3b is going to have this exposed reactive bond, which can then, usually it'll just react with water, and then you have a useless C3b that's just floating around your body and will get degraded at some point. But if the C3b is near a OH or NH2 group, which is very commonly present on the membrane of pathogens, the C3b will then irreversibly attach to the membrane of a pathogen and basically signal to everybody else, like, hey, like, the C3b is showing to the innate immune system, hey, I'm on a pathogen, like, this thing should probably die at some point. And the thing about C3 is that, like, the human membranes also have these OH and NH2 groups, but there's proteins that don't allow for C3b to attach to our membranes. So C3b is only found, generally speaking, on the membranes of some sort of pathogen. Then the C3a, which gets cleaved off, also has this specific purpose where it can just float around and bind to different receptors in the body. So the C3a is called an anaphylotoxin. And this other complement molecule, C5, also creates an anaphylotoxin, which once it binds to their respective receptors, there's some sort of signaling that happens. And from that signaling, you're going to get increased cytokine secretion, as well as more complement molecules being secreted into the blood. So basically, once you have a pathogen in the blood and C3 starts attaching to it, there's this cascade of this anaphylotoxin will then make other innate immune cells create more complement molecules, so more complement can get attached, as well as secreting cytokines so cells in the area can be aware that there's some sort of issue around, there's some sort of pathogen present. And so once you have this complement being attached to the surface of a pathogen, you can then have these complement receptors which can then identify and bind to the actual pathogen itself. And that's what I'm showing here. We have these complement receptors which bind the aflatoxins, which can then further warn the innate immune system. And then you have these complement receptors, which are the ones that aren't green, I guess, and they're gonna bind to all the different types of complement which attach to the membranes of pathogens. Further than that, something that's important to bring up is pattern recognition receptors, or PRRs. And so what these pattern recognition receptors do is that they're on cells in the innate immune system, and they're basically this very general system where you're trying to scan for MAMPs, DAMPs, or PAMPs, which are microbe, pathogen, or damage-associated molecular patterns. So if we're looking at these diagrams here, some of these receptors can identify components in bacterial cell walls, as well as flagellin, which some pathogens use to move around. There's LPS, which is lipopolysaccharide, which is like fatty acid present on, or is it a sugar? I'm not really sure. But this is also where aluminum binds. Aluminum binds these pattern recognition receptors, and that will be important very later on in the video. But also something that's important to note is that generally your immune system is looking for MAMPs and PAMPs, which that's relating to pathogens or microbes, which might hurt you. But then there's also DAMPs, which is damage-associated molecular patterns. For example, ATP. ATP is the energy currency of the cell, but for all intents and purposes, it shouldn't be outside of the cell. So if a cell notices there's ATP nearby, it knows that a cell nearby probably died and there's probably some sort of danger present. So the immune system is also taking care of killing tumors and all that stuff, but that's a random aside. So from that, there's also other types of PRRs that they're looking at proteins, sugars, then also single-stranded and double-stranded RNA, which shouldn't just be floating around randomly. And these are all signs which are indicating towards there's some pathogen present and allowing these innate immune cells to, I guess, be able to signal, create cytokines, and send them out to other innate immune cells and warn them that, hey, there's danger present, we have sickness about. And so just looking at two different cells that are present in the innate immune response, there's more that contribute towards the innate immune response, but I'm just going to mention two. Both of them come from monocytes. So dendritic cells are one of the cell types which can be differentiated from monocytes. These dendritic cells are these really long octopus-looking dudes, which they have these pattern recognition receptors so they can determine whether there's danger nearby. They also have these complement receptors, which these complement receptors are basically just going to bind onto complement, which is bound to pathogens. So these dendritic cells, they're essentially like the Paul Revere's of the immune system, because what happens is initially when you get sick, these dendritic cells are going to bind to the complement on a pathogen, hold on to it, and then run to the nearest lymph node and start interacting with B and T cells so that the adaptive immune response starts to kick in. Because this innate immune response is happening at low levels almost all the time. But once the innate immune system can catch up, that's when you want the adaptive immune response, but it takes multiple days for that to happen. So that's why these are kind of like the Paul Revere's. They're like, hey, there's something that's happening. it's kind of crazy. Like maybe the adaptive immune system would want to kick on, and if in a couple days the innate immune system doesn't handle it, the adaptive immune system is ready to take it over. But apart from dendritic cells, another cell that's important is macrophages, which these are kind of like the garbage men of the immune system. They have pattern recognition receptors as well, but then they also have complement receptors. But instead of just holding on to these pathogens, macrophages will phagocytose, or basically take their membrane and engulf something that has complement on it and then destroy it using cytotoxic molecules, which I won't really get into here. But these macrophages, if they sense danger, for example, with their pattern recognition receptors, they sense that there's a sugar that's associated with a pathogen, they can then release cytokines out around them. And if there's a monocyte nearby that sees the cytokine released from a macrophage, this monocyte will turn into a macrophage and continue and start eating other stuff. So macrophages can recruit other monocytes or macrophages to where there's danger using cytokines. And so I kind of covered the alternative pathway, which that's how you get this C3, this complement 3 attached onto the membrane of a pathogen. But there's other pathways too, such as the lectin pathway, which lectins are types of molecules which bind mannose, another sugar that's found on pathogens very regularly. And there's the classical pathway, which works a little bit later on because it requires antibodies to produce higher specificity for attaching complement to a pathogen. And so looking just briefly at this classical as well as alternative pathway, this alternative pathway, as we saw, C3 can just attach itself onto the membrane, or the C3 can interact with C5, and then the C5 will recruit C6 through C9, which will create the membrane attack complex, which basically forms a pore in the membrane of that pathogen, and the stuff that's on the inside of the pathogen now goes out, which, newsflash, that is not good for cells. And then this is just a little GIF, I guess, showing just visually what happens when you have the membrane attack complex happen. But then you can also have antibodies which bind an antigen, which when I say antigen, this is just a very specific part of some molecule that's associated with a pathogen. So this could be a protein, a sugar, a fatty acid, or some combination of those. So it could be like glycoprotein, whatever. It's basically just some part of a specific molecule that's only associated with that pathogen that this antibody binds to. And when these antibodies bind, they can then recruit the C1 complex, which has I hate this word so much. It can recruit other complements to create convertases. Holy Christ, that's disgusting. But you have these convertases can then convert further complement to be deposited onto the pathogen. And then just going into like a little bit of review, this membrane attack complex can basically cause stuff on the inside of the cell to go out. Then you also have these aflatoxins, which can further warn the immune system that something is wrong. And then you have these macrophages, the garbage men of the cell, are using complement receptors to bind to complement, which was deposited onto pathogens, so that this macrophage can attach onto it and then eat it and destroy it. And so this is what happens in the innate immune response, is this is a response that isn't super specific to any particular pathogen, but it works well enough for most of them. But there's some pathogens that just replicate too dang fast, and you need something more specific that allows for these pathogens to be eaten more fast. And that's where B and T cells come in. So just looking at, I guess, the structure of like what is an antibody, right? B cells create antibodies. This is generally what their structure looks like. You have the heavy chain, the light chain, and then the most important part is this complementarity determining region, just shown in like this light pink there. And these three loops, which are the actual parts that are interacting with the antigen and binding very strongly to that antigen. And so the thing about these antibodies is that your body doesn't know what sequence of amino acids in that complementarity determining region will bind an antigen on the pathogen. So instead of trying to find a very specific sequence that'll work, your body just plays the numbers game and just creates a lot and a lot of B cells, which create a lot and a lot of antibodies, and one of them is bound to have an antibody that binds to this pathogen very tightly. And so that's why you have this innate immune response first, is you don't want to waste your body's energy in making all these antibodies when a complement might be enough, when complement macrophages and this phagocytosis process is enough to kill off this infection. But if that's not the case, you need to make antibodies. And there's many, like when these B cells are making the antibodies, there's these sequences that are in your genome that specifically encode these antibodies. And there's a lot of variation in the types of sequences that can be used to make an antibody. I'm just showing visually here, there's many different types of these V, D, and J regions, which are combined together to make the H chain. And then this light chain also has some variability, which then together creates a lot of variation for the antibody. And that's not even taking account the hypermutation that happens at these complementarity determining regions, which, just a quick aside, these regions are encoded in the DNA. So if you have a hypervariable region, you're just going to have a lot of uncertainty in this complementarity determining region. You're going to have a lot of base swapping happening, which once that sequence is synthesized into the antibody, you're going to have a bunch of random amino acids that are there in the complementarity determining region. And you're hoping that random clump of amino acids in that complementarity determining region can bind to something on the pathogen. And so something you should be asking is why, I guess, or not yet, something else to mention is that there's many different types of antibodies, and I'm not even going to go into that today, but just so you know, there's a lot of different types of antibodies in your body. So IgG is the most common, IgM is the one that's made first in the adaptive immune response, and then depending on the cytokines that are around B cells will determine what type of antibodies they will produce. But as I was saying, why is it that these antibodies don't bind stuff in your own body? So for this, I'm going to talk about how B cells develop in your body. So what happens is these B cells, they grow up in your bone marrow. So once you have what is a precursor to a B cell, they undergo recombination to try and make a BCR, or a B cell receptor, which is essentially just an antibody, but instead of secreting it, it's going to be on the membrane of that B cell. And so this is a deadly process. If you don't recombine the DNA effectively, the B cell will just die, right? And I won't go into the whole recombination of it all because that's going to take even longer. So just let's assume a B cell recombines the DNA effectively and it makes a BCR, or a membrane-bound antibody. I'm going to use those interchangeably. This BCR then is going to undergo negative selection in the bone marrow, in the sense that this B cell is just going to float around the bone marrow for a little bit, and it's gonna try not to bind anything. And if it does, if this antibody binds anything in the bone marrow, it'll automatically be killed. And the reason for that is in the bone marrow, there isn't anything that needs to be, I guess, there isn't anything scary in there. There's no pathogens in there. So you don't want the antibody reacting to anything in there. It shouldn't react to anything in there. So if you recombine and this antibody is binding to stuff that's in your own body, then like that sucks. You're gonna be autoimmune. So your body's like, nah, kill that B cell. So that's what happens. You might be wondering, wouldn't that mean that your body, or you're creating antibodies that only know not to bind stuff that's in the bone marrow? Well, there's actually specific promoters that are in the bone marrow that synthesize proteins from everywhere in your body in the bone marrow, so that these antibodies can have like a mini like theme park of all the proteins that are present in your body, and it has a chance to go to all of them. And if it doesn't bind any of them, that's a good B cell, you know, good boy. It's a good B cell. It probably won't bind to anything that is not a pathogen, and that's awesome. So it's let out into the lymph system, and it's gonna chill out in a lymph node and wait to be activated by a T cell, which we're gonna get to that in a second, but we need to talk about now what T cells do. So a B cell creates antibodies, and it tries to bind to a specific antigen or a region on a pathogen which is distinct to that pathogen, but it needs to get activated by a T cell. And so what T cells really do is they basically are like the scavengers of, they're looking around like a graveyard and trying to find stuff that seems like it came from a pathogen. And so these T cells are going to be scouring these MHC molecules for proteins that don't look like they come from humans. And so these MHC molecules shown here, you have MHC1 and MHC2. An MHC1 molecule will only have proteins that are found in the cytosol of a cell, and MHC2 molecules will only have stuff that was extracellular to the cell and present those proteins. So you have MHC1 and MHC2, right? And the way that these MHCs work is, for example, with MHC1s, if you have some protein on the inside of a cell, it's gonna get labeled for degradation. It's gonna get degraded, and then it's gonna get degraded into shorter bits of protein sequences. So you might have a very large protein, many hundreds of amino acids long, and it gets cut into like 10 to 15 amino acid long chunks, and these get loaded onto the MHC1 and then presented on the membrane of the cell. And then these T cells will go around and they'll sample the MHCs. They'll bind to the MHCs with their T cell receptors, and then they'll hopefully find, if they see something that's related to a pathogen, this T cell receptor will bind even more tightly and make the T cell aware that there is a pathogen nearby, I guess. And the important thing to note about MHCs is that generally they're just gonna have stuff that comes from human origin. So you're basically just presenting stuff that used to once have function, but now is no longer needed and degraded. And you're basically presenting dead proteins that are usually coming from yourself. But in cases where you're sick, if you have a pathogen that gets inside your cell, it'll get degraded, and then it's going to be presented on the outside of your own cell on MHCs, which then T cells can go around and sniff out. And so looking at how these T cells, or the T cell receptor actually binds these MHCs, there's also these co-receptors that are necessary to bind as well. So this differentiates what type of cell can interact with which MHC. So in the case of the cells which activate B cells, they need the CD4 co-receptor, which binds to the side of MHC2. Then you have the TCR in blue and purple, which is binding to the red and orange of the MHC. And that deep red is signifying some protein, which could be of either human origin or potentially pathogenic origin. And so just an interesting note that I'll put in here is that the CD4 co-receptor, which binds to MHC class 2 molecules, is actually what HIV binds to and it gets into T cells with. And so the reason that this is concerning is that since it binds CD4 and gets into T cells and kills them, there's now nothing to activate a B cell. So if you get sick, you aren't going to produce antibodies, even if you have B cells, which have antibodies that can fight against that infection because you don't have a T cell to activate them. And that's how you get, very generally speaking, that's how you get immunodeficiency from HIV through that mechanism. Although it's very likely more complex, but that's just a fun fact I decided to throw in there. Then you also have the CD8 co-receptor, which is associated with CD8 T cells, which I won't really talk about much in this video here. But then looking just at another example of what this MHC looks like, here's like a top-down view of how this protein is basically just lying down in there and being presented to the T cell. Here's another, I guess, image showing this like grapefruit salmon looking color. That's just like some random protein, either human or pathogen origin, that is being presented on this MHC to T cells. And so talking about how T cells are developed, T cells will move from the bone marrow to the thymus and they'll start developing in the thymus. And basically T cells are the modulator of immune activity. So CD4 cells will activate B cells and allow them to create antibodies. And they also modulate CD8 cells, which very generally speaking just kill stuff very effectively. But that's as much as I'm going to get into CD8 cells. But when these T cells are developing in the thymus, what they're making is a TCR, or a T cell receptor, which is very similar to a B cell receptor, except for the fact that you basically just don't have the lower part of the heavy chain, like constant region at the bottom. You're basically just having the top, like, antigen binding region there. There's also other signaling molecules that are associated with both the BCR and TCR, but for time's sake, I didn't delve into that, but just know that it's more complicated than just a BCR and a TCR. But what happens is, similar to how a B cell develops, you need to recombine the DNA to make a BCR. A T cell will do the same thing, where in some cases a T cell will die because it cannot recombine and produce a TCR. Let's say that already happened. Some cells are going to die because they can't even make a TCR. But let's say we have this T cell that could make one. The next thing is that you want to undergo first this positive selection of this TCR should be able to bind to MHC molecules. And depending on which MHC they bind to, that determines which co-receptor they have as well. So either CD4 or CD8. So with positive selection, you just want to make sure this TCR can bind MHCs because that's what it's going to interact with to determine whether there is some sort of pathogen nearby, right? So first, you want this TCR to bind MHCs, but then you undergo negative selection where you want this TCR to bind to the MHC, but you also don't want it to bind super tightly because in the thymus, there's also no pathogens there, right? Very generally speaking, there shouldn't be any pathogens present. So none of the TCR should get super excited about binding the protein that is inside of the MHC or being presented to it. So looking at this image here, just on the very left, we can see this immature T cell is the co-receptor, the CD8 co-receptor is binding, so that's awesome. This T cell is able to bind the MHC, that's cool. It was able to get through positive selection, but the negative selection, it doesn't make it through, because as you can see, the one on the very left is binding way too tightly to what is presumably of human origin protein. So then this T cell is going to get killed. But then you can see the one right next to it, the co-receptor works, that's cool. And then the binding site for that protein which is there isn't very tight, which is very awesome. That's what you like to see. And the reason for this is you want T cells to just go around, bind to various MHCs and just sample them, and you're hoping that the TCR is specific enough for some sort of pathogen antigen such that it can activate a B cell later on along the line. And so now we're actually getting to the part of how a T cell can activate a B cell. So looking at a very, very general image of what a lymph node looks like, once B's and T cells go through development and they're quote unquote mature, I don't know if that's the correct terminology immunologists would use, but you have this B cell area in the lymph node where basically these B cells will go in there and they're going to interact with follicular dendritic cells, which is confusing, but they're not related to the dendritic cells I mentioned at the top of the video. But these follicular dendritic cells, they have a bunch of complement receptors and they're just holding on to different pathogens that have complement attached to them. And they're basically like a Christmas tree with a bunch of pathogens attached to it. And they're basically letting the B cells just look around, just like browse and see if their surface bound BCR binds to any of those antigens that are present. They're being held by the follicular dendritic cells. And let's say this B cell has, like through sheer random chance, it has a BCR/antibody which binds to an antigen on one of these follicular dendritic cells. It'll then it'll phagocytose that pathogen and degrade it. And then these proteins, as we mentioned earlier, are going to be packaged into MHCs, which are then going to be put on the surface. So this B cell is going to be presenting to the world, hey, I ate this pathogen, and here are the sequences like on my membrane, and it's going to be showing that off to everybody else. And this B cell will then go down to the T cell area, and it's going to wait for a T cell to see if it can recognize the peptide sequence or the protein sequence of the pathogen that it just ate off of the follicular dendritic cell. And assuming that this T cell has a TCR that is specific for that sequence of that short protein sequence that was once part of the pathogen, if it's specific enough for that, you're going to have this TCR bind to the MHC. It's going to get super excited because it's binding very strongly, and it's going to signal to the B cell, hey, you no longer need BCRs, you don't need B cell receptors, release these as antibodies. These obviously work very good. This antibody was used to bind the pathogen which you ingested, which I'm now confirming is bad. Don't produce any more surface-bound antibodies and just release a bunch of them into the blood. So this B cell then turns into a plasma cell and produces a ton of antibodies and then releases these into the blood and can then become memory cells, which these are basically just B cells which lay dormant and just wait for the pathogen to show up again. And depending on the type of pathogen you have, sometimes you don't need multiple rounds of vaccination. For example, with polio, you only really need it once because polio doesn't mutate very fast. Whereas with COVID-19, there's a very high throughput of mutations going on. So you need that memory will only last you a certain amount of time until that pathogen is different enough to require more, I think it's a different type of antibody for it to be an effective immune response. But I'm getting ahead of myself. How does this relate to aluminum, which I mentioned before? So what happens when you have this interaction between a T cell and a B cell, which already found its cognate pair, or basically it found an antigen its antibody binds to very well. The B cell found its cognate pair. This T cell is the cognate pair with this B cell because it's also recognizing that antigen. So this TCR is binding to the MHC on the B cell, but then you also require having B7 expression on the B cell, and then T cells will always be expressing CD28. And so what's important is that you need this TCR binding to MHC as well as B7 binding CD28. And B7 is only present on the membrane when you have pattern recognition receptors which tell the cell, hey, there's a PAMP or a MAMP, there's a microbe associated molecular pattern or protein. And once this PRR finds that there's something weird going on, there's something potentially dangerous, it'll then express these B7 receptors, which are necessary for the T cell to activate the B cell. So if this B7 receptor wasn't there, what would happen is that the T cell would just die. And the reason for that, a good way to think about this is imagine you let out a T cell, which is actually autoreactive, and it binds to something that your body naturally produces, right? So you're in the lymph node, and this T cell finds a B cell which has that specific sequence of a protein which this T cell recognizes. This TCR is going to bind the MHC. The TCR is going to be like, hey, that's the protein I want to bind to. This is so awesome. Like, B cell, make more of these. Like, we need to make so many of these antibodies. But if this B7 receptor isn't present, since there isn't any sign of danger around, then it wouldn't be a good thing to activate that B cell, obviously. So then that's why the T cell is killed off. And that is why aluminum or lipopolysaccharide, for example, is used as an adjuvant, is you need to have that pattern recognition signaling of, like, you need to have that signaling happen to, I guess, reinforce in the innate immune system, yes, there is a danger here, which will then allow for these T cells to make B cells produce antibodies. Because if you take, for example, the COVID vaccine, you take the mRNA, which creates the spike protein of COVID, it's making the spike protein, but that doesn't necessarily get registered by pattern recognition receptors. So there's a chance that you're making this protein which doesn't cause any worry for your innate immune system, and there's no B7 being expressed. So even if a B cell recognizes that spike protein and has it on its surface, this T cell could recognize it, but then it's just going to die because there isn't sufficient worry from the immune system that there's anything wrong happening there. So that's the long and roundabout way for why aluminum is found in vaccines, why it's used as an adjuvant. Okay, so editor Kember here. I realized I forgot to talk about something that I think is pretty important, especially given with what's going on right now. I think many of you already know there's been hundreds of measles cases that have been reported recently in the news. And I just wanted to talk a little bit about more of, I guess, why this should be concerning, because Of course, maybe measles isn't that concerning of an illness on its own. You probably won't die. Like the death rate for a measles case isn't very super high, but being contracted with measles is not something you really want in your life. And I mentioned you have a bunch of memory cells, which are basically just retired B cells. They've already done their duty. They've made antibodies that have saved you at one point. And they just like take a nap for a while and they're just around. They wait until they see the antigen again. But the thing that's especially worrying about measles is that not only do you get sick by it, but it kills all of your memory cells. So even if you have an easy pass through with measles, you're gonna have to build a new repertoire. You're gonna have to face all the same pathogens again to create memory cells. So you will have the same immune library that a newborn baby does if you have measles. So that is one of the reasons why being against the MMR vaccine is a little crazy in my opinion, but I just thought I'd toss that in there as another fun fact for you guys. I hope this video was very insightful. And yeah, like I was saying, this was basically just a review from a semester's worth of immunology that I took, so it's by no means very well, I guess, this is just giving you a view. I'm hoping that if you're an immunologist watching this, you're probably not my target audience. I'm hoping that a layperson is watching this and seeing the complexity of everything that you have to think about when making a vaccine, and that this isn't just kind of like we're just injecting stuff into you. Like there's a lot of work that went into this to understand how the immune system works, and it's also still a very up and coming field. It's very recent that we actually realize that how the immune system works, and we still aren't sure very many parts of it, how it works. But I hope you enjoyed. Leave any suggestions below. I'm using my AirPods right now. Hopefully the audio quality is a little bit better. And yeah, sorry about being on the hiatus. I needed to focus on graduating, but seeing as now I don't have a lot of responsibilities and don't have a job yet, I'm probably going to be creating videos more frequently. Make sure to keep an eye out for, I defended my honors thesis a little bit earlier, and I'm going to upload my thesis defense, and then also going to talk further, a little bit more in depth into the science behind what my thesis consisted of. As always, I really hope you enjoyed. Leave any comments below, like if you enjoyed this video. Have a good one.